Hello everybody, my name is Ruben Hermann and I'm a PhD student from the University of Constance. I'm happy to show you part of the results which I have on my thesis, which is on indirect evolutionary rescue in a predator-prey system. When a population is faced with a perturbation, this can lead to a strong decline in population size, maybe even to extinction. But within trait variation of the population connected to trade-offs can buffer the perturbation, thus minimizing its effect on the population. A mechanism for that is evolutionary rescue, where a threatened population adapts to environmental change that otherwise would have caused its extinction. An exemplary graph for that is here, where a population is faced with environmental stressor, leading to a strong decline in population size, which could lead to extinction. But during this population decline, the population has time to adapt to the environment through either, for example, a genotype which is already present in the population, or for the mutation of a new resistant genotype. And with the increase of the frequency of the new genotype, the population can again have a positive growth rate and thus recover from the environmental stressor. By having a higher genetic variance in the population, this can of course lead to faster recovery and thus a higher likelihood of being rescued from the environment. While this has been already thoroughly studied in a single species in modeling and in experimental setups, there's still a lack of knowledge on how this might work with communities where multiple species exist and interact with each other. A possible mechanism there was described in a modeling paper by Yamamichi and Miner, where a threatened population is saved from extinction through the adaptation of a non-threatened interacting population. For example, we have a predator-prey system where we have a single predator and a prey population consisting of an undefended genotype and a highly defended genotype which is connected to a trade-off in the growth rate. If now the environment changes, leading to an increased mortality of the predator, leading to a decline in the predator population size, this leads then to having a lower predation in the system, and thus the undefended genotype outgrows the defended genotype of the prey population. But having more undefended genotypes available in the environment, the predator then is then rescued from this environmental change through the adaptation of the prey population. While they have investigated this in evolutionary rescue, there's still a big lack of knowledge missing when this might occur in an environment. So I want to jump into this and investigate further when evolutionary rescue occurs. For the start, I want to use a mathematical model where I can use a range of mortality rate for the predator and varying frequencies of the two different prey clones. With this, I will be able to predict different evolutionary rescue events. And once I have the results from the model, I then want to use those to implement them in an experimental setup. For the model, I use an adaptive model from Bex et al., where I can simulate two different clones, one being again highly defended with a low growth rate, and one having a low defensive connected to a high growth rate. I have as well a single non-evolving predator, and I will change the parameters of the frequency of the different prey clones and manipulate the mortality rate of the predator. When I start the simulations, I first have to define when evolutionary rescue happens in my model. So at the start, of course, the predator has to go extinct when the prey population is not able to adapt to a changing predator population size. Secondly, I have to have this typical U-curve where through the environmental stressor, the population size of the predator declines and then increases again, which is connected to a change of frequency in the prey population. Going to the results, here I have on the y-axis the frequency of the defended prey population, of the defended prey clone at the start of the simulation. On the x-axis, I have the mortality rate of the predator. Here we see the results for that. On the blue squares, the predator are able to survive the perturbation without the evolution of the prey population, and the white squares, the predator population goes extinct even with evolution of the prey population. But the sweet zones are the purple squares, where the predator would go extinct if the prey population would not be able to adapt, but does survive with the evolution of the prey population. With these results, now I will go to the go back to the lab and start experiments to see if I can reproduce this also in the lab. For that, I'm using the predator-prey system consisting of the freshwater rotifer Brachyonus calytiflorus. And for the prey population, I have the green algae Chlamydomonas canhati. And I have an undefended clone, which is here depicted as a single cell. And I also have a defended clone. The defended clone, for example, then can clump to cell clumps which are 16, 20, or even more cells clumping together in one big clump, and they are then too big for the rotifer to ingest. At the start of the experiment, I always have the same density for the predator and for the prey population. I only change the mortality rate of the rotifer through adding sodium chloride, from now on just called salt. And this adding of salt does not impact the growth rate of the prey population. I, as well, at the start of the experiment, can then again change the frequencies of the defender algae clones. For the results, here on the x-axis, I have the different salt concentration. On the y-axis, I have just different starting frequencies of the defended algae clone. On the z-axis, I have the growth rate of the rotifer depending on those two components. Now here we see at a low salt concentration, the rotifer has a strong growth rate, so they don't go extinct in any of the different frequencies of the defended algae clone. But having a high salt concentration leads always to the extinction of the rotifer. If we now add a plane to that, then we can nicely see how also having higher frequency of the defended algae clone leads also to a stronger decline in rotifer growth rate. And the most interesting treatment here is the 5 millimolar salt concentration, as we have here partly a high rotifer growth rate. And when we have too high frequency of defended algae clone, this leads into extinction of the rotifers. So now I want to look deeper into this treatment. Here we have the survival rate of different rotifer populations. On the y-axis, we have the survival of all of the 10 replicates of the rotifer population, and the x-axis different treatments for the rotifers with a different starting frequency of defended algae clone. We see if you have only defended algae in the prey population, the rotifer always goes extinct. But if you have too few to none defended algae in the population, the rotifer always survives. The interesting are the middle areas where part of the rotifers are surviving and part are going extinct. And the most promising treatment is the one where we have 70% defended algae in the start of the experiment, because there we have approximately 70% of the rotifer population surviving and 30 going extinct. If you look now more detailed in this treatment, we see now the time of the experiment, which was 8 days. On the y-axis, left-hand y-axis, we see the density of the rotifer population. On the second y-axis, we see density of the algae population. At the start of the experiment, we have a higher frequency for the defended algae clone. This, connected to the salt, leads to a decline in the predator population size, and the lower predation there leads also to an increase of the undefended climate clone and to a decreased abundance of the defended clone. Having more undefended algae in the environment leads then to an increase again of the predator population, going back almost to the same density at the start of the experiment, and thus recovering from the perturbation. The increased predation again then leads also to a decline in algae density from the undefended clone. And here we see nicely how we define indirect rescue happening in the experiment. So concluding, I was able to show a range where indirect evolution can happen in a model, and then prove that this happens in experiments as well. And with that, I can show indirect evolution is a mechanism that can work to rescue a non-evolving predator through the adaptation of its prey. And I was able to show in a model and experiment that at which combinations this can happen, so in which range indirect rescue can happen. With this, I want to thank you for your attention. I also want to thank my two Hiwis, Natasha and Tamara, who helped me a lot. And of course, I want to thank a lot Lutz Becks, my supervisor, who helped me a lot through all my work, and my group for answering, of course, many questions for me. And I hope you have many questions.